Thank you so much for the great introduction and having me here. So a few words before I start. This talk will feature exactly one joke and one competition with prizes. Ready. Ask questions whenever you want. We're going to keep it informal, and this is for all of our enjoyment. And since I'm being recorded, that gives you an amazing opportunity to ask me something embarrassing, and that will be captured on YouTube. All right, so without further ado, I want to start with talking about the importance of data. I want to give an example outside of our field, and maybe let's start with a raise of hands of who roughly knows the story of discovery of the DNA model. All right, it's good. So basically, it's actually a controversy who should take credit for it. There have been several scientists working on it, including Crick Watson and Maurice Wilkins, as well as Rosalind Franklin. And as you probably remember from who got the actual Nobel Prize, Rosalind Franklin wasn't among them. And that's because she unfortunately died of cancer before that happened. But the controversy is not about the circumstances of her death, but it's about how the discovery actually happened. There was a different set of independent scientists who acquired the data and who interpreted the data. So this photograph on the left is a crystallography depiction of one of the forms of DNA. And this photograph, named photograph 51, fun fact, someone actually made a theater play about this whole story called Photo 51. And this photograph was instrumental to actually figuring out what is the model of DNA. And it turns out that this double helix composite was the key to how it replicates. And there are different interpretations of the story. As I said, there's a lot of controversy. It was a long time ago. We have also issues of gender playing here. But the main take-home message I take it from it is that we can have situations where one set of researchers, group of researchers, acquires data, and a completely different group of researchers are able to perform a breakthrough discovery using that data without being directly involved in the acquisition process. And we see that in other fields. Let's start small. If you look at the statistical maps deposited on neurovault.org, we see more and more papers using this data to do quantitative comparisons of spatial patterns in the brain between the published studies and their new discoveries. And we have shown in another paper that such comparisons, when you use unthresholded maps or minimally thresholded maps, is much more precise than using any sort of high thresholding or just coordinates or just basically eyeballing figures on other people's papers. If we go a little bit bigger and we look at OpenFMRI, we did this study a couple of years ago when we look how many people actually reuse data coming from OpenFMRI. And depending on how optimistic you are, you can see there's either linear or quadratic trend. Nonetheless, we see a growing number of reuses, and that was the analysis was done in 2014. And at that time, we estimated that if all of these studies had to acquire all of this data that they used to publish their science, that would cost around $800,000, $900,000. Right now, it's definitely above $1 million. So we see that sharing data not only enables science, but also saves all of us money. If we look further, we can only see that we can also see that studies sharing data have higher statistical quality. So in this particular paper, what they looked at is whether there's a difference in statistical quality of papers, looking at statistical errors in the paper between between papers that express willingness to share data and those that did not. So this is the classic case of like, I'm going to share my data upon request, and then actually someone requests the data, and outcomes can be surprising. And then finally, what we also see that sharing data is related to higher citation rates. And this, of course, this is all correlational evidence. But what we see is that papers that include data sets are cited more often, maybe because they provide more value, maybe they're because they are of higher quality. But both of those things could be translated in quality of science. But those are all aggregations and basically studies on different groups of papers. But there are also examples in our field, in neuroimaging, in human neuroimaging, of interesting and important papers that have been performed using publicly shared data. And those are just a few of them. And maybe some of them are more useful than others. Some of them are actually quite old. That Mike Gracious paper was using a database that doesn't even exist right now, the fMRI-DC. The cluster failure paper, we might not completely, all of us, agree on the outcomes and results of this paper, but nonetheless, it is an important discussion to have. And that was possible because of shared data. I actually don't believe that Anders ever acquired any data. So basically, this mode of operations enable new type of science and enable us to be more efficient, but also start asking questions that otherwise wouldn't be asked. So when I think about all of this, and I'm trying to ask myself the question, how can I or us make the biggest impact on science? I can kind of boil it down to one sentence: making more data accessible to more researchers. And there are different aspects to this sentence, basically what is more and what is accessible. But basically, this is kind of my modus operandi. And during this talk, I will try to walk you through some of the tools and some of the approaches that we are trying to work on to achieve this goal in various ways. And I hope that you can tell me where we're wrong and where we're right and how can we improve. So I'm going to start this by talking about the data and how we all describe the data, especially in and to tell this story, I need to introduce you to Professor Smith. And Professor Smith is a young, up-and-coming PI with many ideas and a dedicated lab. And she just came up with this new study that she could do to look into attentional processes. Instead of collecting new data and spending more money, she would reuse a data set collected in her own lab. So there's no data sharing involved. It's basically data that was collected in her own lab a few years ago for a different question and a different study by one of her PhD students, Mike. Mike, since he graduated, decided to become a data scientist in California and is unreachable. Basically, he doesn't pick up his phone, and we don't know what happened to him. Since he was involved in this project and he was the sole owner of it, he basically ran the show himself. And when Professor Smith looked at the data that she found on some external drive lying beneath a stack of books, she discovered basically a mess that she cannot make much sense of. And that happened in her own lab. This is not some elaborate collaboration between scientists speaking different languages across the ocean. It's something that can happen within the same lab. And probably to a certain extent, you can see how that is happening not that far from us. So why is this really a problem? Well, this heterogeneity in how we treat data makes a lot of sense to us when we do it, but later it can provide problems in sharing data within or between labs, and also causes unnecessary manual input of metadata. When we analyze the data, we have to figure out what are the different scanning parameters, et cetera, and whenever we have to do that by hand, there's a room for error. There's also no way to validate consistency and completeness of a data set if there is no standard that we all agree on. But it shouldn't be that hard. We have been using MRIs for a while, and there are 3 or 4 vendors that produce MRI scanners, and they all produce data which is roughly of a similar format and a similar kind. There's also quite a few similarities in experimental designs, or at least you can categorize them in certain ways. So there should be some common way of storing and describing whole data sets. So to fill this gap, together with Satra and other people also in this room, we got together and tried to involve the whole community to build a standard for describing data sets to avoid such problems and also make it easy for people to use tools, which I'm going to talk about later. Basically, who is it for? It's for the lab PIs, versus Smith, for example. It's also for workflow developers, so tool builders, people who are designing methods for other scientists, and they want those tools to be as easy to use as possible. And finally, database curators. So we see more and more of those repositories where you can upload your data. When you do that, those repositories care very much about the quality of your data sets. And now when there's a standard, they can validate the consistency of your data set. And finally, it is for you. Because it will enable you to use a big and growing set of different tools in a much easier and less error-prone way. And we have a few principles when we try to come up with these standards. We really cared about adoption, and we spent a lot of time reaching out to different scientists in different labs, computer scientists, cognitive scientists, physicists, etc., etc. We also didn't want to reinvent the wheel. We didn't want to force people into vastly new file formats. And we didn't want to spend too much time doing something that has been done before. And we also wanted to focus on those parts of neuroimaging that produce the most data, at least in the beginning, to address the needs of the vast majority with the least amount of effort, and then basically fill in the gaps. That's why, as you probably see, the standard is vastly focused on MRI data, and only now we are going into MEG and PET data. There's a whole process involved. We had a kickoff meeting, we had hands-on meeting during GeorgeBM, then we presented it at a specific neuroinformatics conference, and finally there was a release candidate, a public call for comments. Hundreds of emails were sent, and I had to go through all of them. And for a certain period of time, I could not look at my mailbox. So lots of fun if you really like emails. Finally, we reached a lot of people for comments. Some of them actually replied. And we also produced a lot of example data sets. That was crucial to basically not only talk about this abstract thing, but also give people examples and show them, like, this is how a BITS data set looks like. Do you like it or not? Would it be hard to build a tool that uses or not? And to kind of quantify how many people were involved in this, we had, I think, 27 co-authors on the final manuscript. And that's another fun thing. Editing a paper with 27 people. Great, especially when, yes, okay. But it was totally worth it because we have our standard right now. And it is following very simple principles. So one thing that we discovered very quickly is that in many, many labs, people are not using sophisticated databases with APIs and things like that, but relying on formalized and sensible structures of files and folders. And for those with a computer science background or inclination in that direction, that might seem rather Stone Age, but it actually makes a lot of sense. It is very practical, especially when you consider how neuroimaging software that we will use, FSL, SPM, etc., how this software actually takes data in. So we were to go and base all of this on some kind of a programmatic interface or a database, all of the software had to be adapted, and that probably would kill the whole project. So we have a structure of files and folders, which is quite formalized. You can see there's a split for subjects. There's also an optional level for sessions, so longitudinal studies are supported as well. We use tab-separated files for all of the tabular information, for example, demographics and things like that. Finally, we use, well, not finally, but we do use Nifty for the binary imaging data. This is the most common thing that is used out there, and that's why we've picked it. But I can tell you, when I went to Montreal to talk about this, there was a lot of discussions basically about how the format they've developed a while ago, Mink, is far superior to everything else. Who knows Mink? And I agree, it has a lot of good features, but unfortunately, adoption is something you have to take into account. And then we used JSON to store key value pairs, so basically metadata concerning scanning parameters, etc., etc. And this is also a good time to talk about the fact that we really focused on capturing and defining these types of metadata that are crucial for analyzing datasets. It wasn't just trying to have like as complete description of the dataset as possible, and that would take us a very long time, but really wanted to be practical and kind of ask the question is, do I know enough about this dataset so I can give it to someone else and they would be able to analyze it? So this was our driving force. And then all of this would have been just a very, very boring document with many pages that no one will ever read and would be very hard to comply. So we also built a tool that you can drag and drop or actually point to your folder on your hard drive and validate your dataset. And it will tell you whether it is a valid bits dataset or not, and if it's not, it will tell you what are the different incompatibilities. And that tool works either on the web as well as there's a command line version of it. And it doesn't share any data, even if you run it from the web. All of this validation is happening client side, so there are no concerns about privacy and IRBs and other scary things. Okay, so we can go back to Professor Smith, who, after learning a certain lesson and eventually figuring out how this dataset looks like and publishing upon it, but wasting quite a bit of time, in the future, we'll be able to adapt certain community standards to avoid dealing with the mess of data. Well, but I also promised you to talk about the tools and how around bits there's also an ecosystem of different tools. And this is not an exhaustive, but somehow exhaustive list of different tools that were built for bits, and many of them are converters to basically go from bits to from DICOM to bits and things like that. Some of them are quality assessment tools. There's also fMRI prep, which Chris over here is developing, which I'm going to talk about soon as well. But one of the driving forces, at least for me, is that now that we have the standards, that we have a lot of data sets complying to, it's much easier to write tools that you can just point to a data set and it will just run. So we wanted to make this happen, and we wanted not only the few tools that we develop in our lab to be able to understand bits. We, again, wanted to get the community involved, and we wanted to make this whole process even easier. So we came up with an idea called Bits Apps, because in Silicon Valley, everything needs to be an app. So that's why we called it this way. And the idea is combining two aspects. One of them is that those apps, which are basically portable neuroimaging workflows do understand bits data sets. So basically, you point them to any bits data set and they run. And the other aspect of this project is that those apps also are self-contained. So in other words, they include all of the binary dependencies with them. The same way when you open your phone and you install Flickr, or does anyone use Flickr anymore? Okay, Yelp. Okay, I've heard that Yelp is the app for very sensitive people. Nonetheless, yes, you install an app and no app will ask you, oh, do you have version of this glibc library installed? Or like, what version of FreeSurfer do you use on your iPhone? No, it's all easy because all of those apps, if they have any dependencies, those dependencies are included inside them. So why don't we do the same thing? Yes. So anyone trying to deal with all of these dependencies, especially in context of supercomputers and things like that, it's a bit hellish, especially when you have to deal with other people maintaining your versions and things change just in that middle of your analysis. When you finish analyzing your controls, someone upgrades FreeSurfer to a new version, and then you started analyzing your clinical population. Suddenly you find a found, or you didn't find it because no one told you it was upgraded. So all of these things happen. So there's not only convenience, but there's also a possibility of additional confounds. So how are we going to deal with this? I'm going to use a technology called containers, and I'm going to have an attempt to make this section exciting, but I might fail. So I apologize in advance. So what are containers? Basically, it's a big bucket where you put all of your binary dependencies. It's a little bit like the whole operating system in one single file. You probably heard about virtual machines. Who heard about virtual machines? Aye, yes. It's a bit of an older technology, and virtual machines also perform the same goal. But there's a slight difference between containers and virtual machines. Basically, virtual machines simulate all of the it's actually closer to computational biology or computational neuroscience than you might think, because all of the hardware and its calls have to be simulated. And on top of that, you have the software and the operating system. So simulating all of this hardware is usually time-consuming and can provide, introduce performance issues. But containers share with the host, so the computer we run it on, much more, and they basically share the kernel and the software that is emulating all of this. So running containers doesn't have to simulate hardware, and things are faster and smaller. And some of you might have heard of Docker, and Docker is one of the implementations of this container technology. And it consists of two things. There's a Docker engine, which basically runs the containers, and there's the Docker hub, which is a centralized repository of all of these images. And this is quite important and very useful, because now when we have those apps, we have a place to put them. And that place is actually hosted by someone else, and we don't have to deal with this. And the other good thing is, which I'm going to come back to, is that we can store all different versions of our software. So it's very easy to go back and forth between different versions for different projects, for example. But Docker has some limitations. It basically is designed for the cloud. It has and that has certain implications in how it was developed. It has some dependencies that provide security concerns on systems that have multiple users. For example, clusters. So I presume many of you use the cluster that Satra is running. Does it have a name? Open Mind. It's very progressive. When I asked this question at Berkeley, and they asked them how do they call their cluster, they said, we call it cluster. So there you see the difference in redundancy. Okay. Okay, so there are concerns in privacy when running Docker, and it's almost impossible to get Docker installed on high-performance computers because of those concerns. But it turns out that scientists don't actually care about all these features that Docker provides that are for web applications. So I'm going to skip this a little bit. The bottom line is of this extremely exciting story that there's another technology that we use called Singularity that allows us to convert Docker images into something else and run them on high-performance computers, which is kind of cool because you can use Docker to create an image that captures all of your environment on your laptop and then run exactly the same thing with the same dependencies, same custom versions of whatever you need on the cluster on 1,000 cores and 1,000 subjects. We had some other decisions concerning parallelization, which I'm going to, if you are interested in this, I'm happy to go back, but we are running a bit short on time, and I want to have some time for you guys to ask questions. But what is important, we, again, engage the community. We run a workshop where we invited something around 30 people, some leading developers in the field from representing software such as FSL and SPM and CPAC and things like that, to take their existing workflows and their apps that you guys know and love, and first of all, make them compatible with bits, and secondly, make them run into a container so they will be easy to install, as easy as Yelp. All right, so the outcome of this are those beautiful 22 different bits apps right now, and hopefully that number will be growing. It's a bit of an eclectic set. You probably recognize some of those names. We have SPM here. We have FreeSurfer, we have Broccoli, which is this GPU accelerated preprocessing and statistical analysis toolbox, automatic analysis, NIAC, CPAC. There's plenty of it. And that diversity was very important for us because that's a test for us, whether can we accommodate all these different pieces of software written by so many different people with so many different ideas and philosophies, and also using different languages. We have MATLAB here, we have C, we have Python. I mentioned versioning. Each of these apps, when they're released, when a new version is released, there's a new Docker image pushed to Docker Hub, and all of those are stored there for you to use, so you can switch between them, which is very useful and improves reproducibility. Okay, but enough theory. Let me introduce you to some of the apps that we are working in our lab, in our center back at Stanford. And the first one I want to talk about is MRIQC. So this is a tool for doing quality control. So basically, when you scan your subjects, and it's both for anatomical and functional scans, what it will do, it calculate a wide range of different quality control metrics. Some of them more common, for example, how much the subject moves, using estimating that using frame displacement, or how smooth the image was, and some of them more obscure. We try to be very inclusive in terms of different metrics that we use, because there's no consensus of which of those metrics can pick up on artifacts and outliers. And if you collect enough data, you can figure out what's the distribution for those. But on top of that, we also provide easy to view and quickly evaluate reports. So for every run, either anatomical or structural, you're going to have a neat HTML file, which is, by the way, self-contained again. So you can take that page and email it to your radiographer saying, like, what is this? What happened there? And you can use those reports to evaluate quality, or basically spot an outlier on one of the metrics, you zoom in and try to figure out what happened there and whether you can improve the quality of your scans later. So we plot here, this is the mean EPI. We also plot the standard deviation map. And this is the promised competition. So there is an artifact on this image. And whoever first spots the artifact, I will buy a drink tonight. Folks at NIH got it. No pressure. Allowed to say anything. I can give you a hint. Oh, was that a hand raised over there back? Okay, slice 56. So that's this one, yes? Yes, it's there as well. Yeah. So what could you tell us more? What concerns you there? '56. Sorry, this one, yes? Oh, you don't see what I'm pointing at. This is great. Okay, I can move. That's great. So you're saying that this one is brighter than this one and this one, yes? This is not it. This is not it. It's something else. Well, you see it, you're going to be like, oh. This is a multi-band sequence. Okay, there's another theory.
Smaller slice.
You mean this or?
Starts around 40.
Okay, yeah, this. It's related to that, but it's much, much worse.
That's right.
It's still not it. The standard deviation map. 24 to 26, yes. Sir, you want a drink. But actually, collectively, you're all three want a drink. Whoa. Okay, that was the gong. Well, actually, you are on the same track, so I guess I owe both of you a drink. So yes, what you can see here is that variance from the eyes, which should be contained in those bottom slices, is leaked across slices and back into the brain. And you can see it here inside the white matter. But what is also interesting, you see a bit of it here as well. So you sort of almost got it. So yeah, multiband, huh? So that is a problem still that we are trying to figure out with the reconstruction we're using, which is exacerbated by the fact that people are using their eyes in the scanner. We never predicted that. Basically, blinking and moving your eyes makes that worse, and there are other. We can talk about this. It's actually quite interesting, but it shows that these sort of things can help you figure out and see these problems. All right, there's also a video on YouTube I've posted with this artifact, and the white matter is blinking at you. It's super creepy. We also show these time series plots, which include different time series. The first one is signal in the air on the sides of the brain. So there should be nothing there and no meaningful variance unless there are some problems with the scanner. Then we have a percentage of outlier voxels calculated by AFNI. And then you have DVARS, which is basically correlation with the previous volume. And then motion measured by frame displacement with an arbitrary cutoff of 0.2. And finally, we have a carpet plot, the same thing as Jonathan Power was using. Who knows Jonathan Power carpet plots? The whole paper about a plot. Yes. Right, you know it. All right, excellent. All right, so this is it. Let me show you how it works in action. So one of the artifacts that we are concerned about is white pixel noise. In other words, it can be caused by electrical discharge when you basically have a metallic object that moves in a constant field, physics, and then it touches another metallic object and there's a spark, and that spark causes a radio frequency pulse, and that pulse interrupts image reconstruction by causing a local intensity in the K-space. So that means you're going to have an artifact specific to a certain frequency. So this is how this very severe case of this looks on those plots. You see there's normal motion, they move their head a little bit, and then you have this. And the interesting thing about this is that you see it in the air. So remember the top graph is the air beside the head. So there's no ghosting, none of this like front and back ghosting. And if there is a difference in signal, that means there's something wrong. So you can go back and actually look at this kind of what happened there. Another interesting thing that we can spot is influence of breathing on the signal. So as you can see here, not much happened. Motion is very minimal. There's maybe some, but then you see this increase in outlier count over there. And if you look at the carpet plot, you see a decrease in signal that goes on for a couple of seconds. And a similar, slightly smaller thing happens here. And this is where a participant basically took a deep breath. And that can influence the bolt signal. The scary thing is that all of this will suddenly correlate with each other. So there you go. Now is the time for the joke. Are you ready? So while I was preparing a class about quality control and artifacts and things like that, I asked Twitter, send me your artifacts, send me your words. And Twitter responded with this. So I could not figure out what's wrong with the picture on the left. But the one on the right is a funny story. So let's sync. All right, so the one on the right is actually a hair product that had some metallic elements in it. And that caused a disruption in the field. And basically, it looks like this on the scanner. Since I saw this picture, there was a case study published in ISMRM or something like that about a very similar case where there's an interference caused by a hair product. Okay, so that's all about the MRI QC, which you can all use and download and install and using different ways. And I'm going to put all the links at the end. But the next tool I want to talk about is fMRI Prep. So fMRI Prep is a simple fMRI data preprocessing tool. And when we say preprocessing, we obviously mean something else than everyone else because no one ever agrees on what preprocessing is. And by this, we mean a simple denoising and normalization. So there's nothing to do with fitting models, which you can do in so many different ways. Doesn't do DCM, doesn't do connectivity or dynamics, and all these different things. Just takes all of your subjects and does motion correction. Extract different regressors of basically confound regressors and puts them in the same space. And does this robustly, basically on any data you can throw at it. We obviously may want to make it easy to use and install. And the robustness is key here. It's not a pipeline developed for a specific scanner and a specific sequence just for this one thing. We really want it to work on many different data sets. And the way we're trying to achieve that is by capitalizing on OpenFMRI with all of the different diverse data sets from different scanner, different resolutions. We're basically testing this on many different types of data and see how well it works. We also want it to be robust, so kind of click and find, but we don't want to lead to kind of decreased awareness of how science works. We want people to be able to understand what happened to their data. So we also provide documentation that guides people through the process and lets them answer questions during conferences, for example. What did you use for motion correction? And it's like, I don't know, I pressed a button. Want to avoid that situation, and they can basically say, I use this tool which uses this thing, if they so wish. So we're doing T1 preprocessing, bias field correction, use ants-full skull stripping, which works beautifully basically all cases I've seen. And then tissue segmentation using FAST. And then we have this twist on MNI co-registration, also using with a robust set of parameters that we iterate through until we find something that works. Then using motion correction using McFlirt, which soon to be replaced by ANTs as well. And then for skull stripping of APIs, we're using NiLearn with a stick. And finally, for EPI to T1 registration, we're using FSL Fast, which also works quite robustly. But right now, we've been testing this for a while, but we also don't want to force everyone to trust us that this data was analyzed in this perfect way, even on data that we never seen. So we also put a lot of effort into reports. So the same way as MRIKC have its own reports for quality of the raw data, this tool has reports for quality of the processing. So you can go and verify whether the tool did what it promised. So I'm going to show you how one of these reports looks like. Oh, this is spoilers. OK. So this is one of the reports. If you had more runs, you could basically go to a different run. So there's one report per subject. And here you have a picture showing skull stripping. And then you have segmentation. And this is all automatically generated for every subject. And here you have co-registration. And the cool thing is it moves. So basically, it blinks in and out between the MNI template and the subject's T1. So you can see how well the co-registration was performed. And when I go out, it stops blinking. Then we can ignore this for this example, but here we have anatomical CompCore. So we extract signal from different anatomical regions that should not have meaningful bold signal. So ventricles, white matter, and things like that. And we provide all of those signals for you to use in your models. We don't make the decision of we decide to regress this out or that out. No, we give you the regressors, and you can include this in your model and decide what sorts of censoring you want to do and things like that. We give you the tools to do it, but there isn't enough consensus in the field to basically say this way or another. It's actually even hard to cover all of the different schemes people like to do. And then we also have co-registration between EPI and T1, and this one is using the boundary. So we plot the boundary, so you can see how well that works. And here you have skull stripping of the EPI. So this allows you to quickly evaluate how well the preprocessing of your data was performed. OK. Back to the slides. And also, we try to make the output all neat and named in the right way. That kind of loops back to one of the goals, and hopefully the whole vision will be much more easy to understand soon. But when I said making more data accessible to more researchers, part of this accessibility is also providing data in a form that enables more science and enables the science to happen quicker. So as we argue in this paper, there are different forms of data sharing. If you put peak coordinates of your activation in your manuscript, this is a form of data sharing. This is a machine-readable piece of data that people actually crawl and do meta-analysis on. You can upload your maps to NeuroVault, or you can put your raw data into OpenFMRI. And all of these things are kind of on the increasing scale of reusability. And if you want to go a step further, you can also provide your data preprocessed. So that's why we're investing so much effort into this tool, because we want to also provide all of the OpenFMRI data set in a preprocessed form that will be much easier for people to just grab and answer the burning scientific questions they want to ask. But on the topic of making more data accessible to more researchers, some of you are probably curious why I'm talking about all these tools and all these standards that all can be used by individual researchers, and that actually might not lead to any data being shared with anyone. I mean, you can take bits and implement in your lab and decide not to share your data with anyone ever. You can take any of those bits apps and install them and run them in your lab yourself. And again, how does this actually promote data sharing and makes all of this open? Well, one of the reasons we invested so much time into those efforts is because we are building and soon launching a platform that provides science as a service, as a free service for everyone. And it capitalizes on the bit standard and provides all of those bits apps for you to run on your data. And the reason why we're doing this is because it is an incentive for data sharing. So when it will launch and after we'll go through the beta testing period, there will be a requirement for making your data publicly available after a certain grace period in exchange for using the platform. So I can briefly attempt to demo it to you right now. As you know, all demos break. So the platform is going to be called Open Neuro, and it's basically an extension of OpenAI. As you can see, it's very pretty. I did not draw this. We have very talented people in Portland who made this very pretty. You can basically log in with your Google account. And when you log in, you're going to see all of your data sets. And I obviously uploaded a bunch of things because I've been testing it. But let me show you how the upload process looks like. So let's say you want to use the platform. You have your data set. You use bits to organize it. So you can go and choose a data set. Let me just choose this one. And validation just finished. This is a multi-gigabyte data set. And one of the things that we put a lot of effort into is not to waste your time. And we achieve that by validating the consistency and of your data set and whether it is truly a bits data set before you spend hours uploading it. So this is all happening client side, and here you can see whether there are any warnings. For example, here I didn't have a slice time for this data set. It's a very old data set. That's actually the first data set that was ever submitted to OpenFMRI. And for the love of God, we don't know what is the slice time for that data. But when you upload yours and you don't have the slice timing, you might be able to recover it. Okay, but this is just a warning. You can still do analysis without slice timing. You're just going to skip that step. So I'm just going to press continue. And the data set is here in the background. So that's great. I'm not going to show this right now, but we've built in all these things where you have interrupted uploads. So something happens, you can just resume it. Your laptop dies, you buy a new one, whatever. But this is just working with data sets. So let's have a look at one, which I think demoed at NIH. So that looks like every other bits data set, but you can still modify it. You can add a new subject, you can change a piece of metadata, et cetera, because it's a live version of it. It's a draft. But let's say I would like to run some analysis on it, because that's what the platform is for. I press run analysis, and it will ask me to create a snapshot. So what on earth is a snapshot? Basically, we believe that we want to have this trace of breadcrumbs between the results and the original data set. So we have the results of your analysis. We have a container, which is a mutable frozen piece of software that was run on your data set. And we also need to have a frozen state of your data set. You can't just say your data set at any given point. It has to be your data set at that particular point. So that's where we're going to take a snapshot of it. The snapshot is actually very lightweight. Also put lots of effort into making this fast. So even though it's a multi-gigabyte data set, no copying is happening when you create a snapshot. And then you can create run a task. So let's say I'm going to run this. You can choose a version, and this eventually will include all of those bits apps. So you will be able to run all sorts of analysis, and we hope that it will grow in the future. You can change some parameters if there are any. For example, I can restrict this to particular subjects, and I can press start. And of course, we're going to break because it's a demo. No, we didn't. Okay. So what happens in the background that your job is being sent to Texas or Palo Alto, and it's being run on one of the high-performance computers we work with. And then when it's finished, because if I just click OK, and if we have a look at it, with staging input, which basically means copying data. When it's finished, you're going to get, or actually I'm going to get an email saying, oh, it's finished, go look at the results and start writing. I can also quickly, how are we doing in time? Well, so this is basically you can then browse and download your results. We have facilities to do a few things online. So you can, for example, open Nifty files and browse them on the platform. And this is something that we are working with, and this is a way to provide something in return, some incentive for your generous gift of data. And this is how all of this culminate together. Okay. Just in case it didn't work, I had some slides which I'm going to just skip through. Okay, since I have exactly 3 minutes, I can talk a little bit about the future directions we have and things I want to explore with this platform and with these tools. First one is something I call hypothesis generation machine. So when you think about having data described in a formalized way, and you have both of the dependent variables and the independent variables. So you have the behavioral metrics, and you have the raw data that you can use to extract different brain signals. You can replicate the analysis that this data set has been used for. That's one thing. But more exciting, you can use the same data to test things. Or in other words, you can go on a massive fishing expedition. And now you can ask, like, what's the value of a fishing expedition? What's the value of this exploration? Like, none of this could be treated seriously with the same scrutiny as a truly pre-registered, hypothesis-driven test. And you're absolutely right. But if we find a strong relationship on this exploratory mode, and let me give you an example. Let's say someone acquired a data set to do the classical task-based mass univariate analysis. And they answered their question, they've published a paper, they've made the data set publicly available, and moved on to different questions. We can take this data set and now ask different questions. We can see whether the particular paradigm, particular contrast, particular cognitive process they were studying, whether it also involves changes in connectivity, whether there are differences in the segregation versus integration of the networks involved during the task versus off the task. All these questions could be asked later on. If we see strong relationships, this will generate a hypothesis. This will allow other people to say, there's something to it. I should acquire more data to figure out if this replicates. Another thing that we are interested in is any of you doing imaging now, there's a lot of seemingly arbitrary decisions you have to make to get through an analysis. Starting from, for example, a smoothing kernel and decision which software to use, which regressors to add and which regressors to omit. And we don't have the resources, either in terms of time or computation, individual researchers to assess how much of a difference those decisions actually make. And there's quite a lot of concern in different fields of something called vibration ratio, so ratio between the highest and the lowest effect size when you iterate over and sets of decisions. There's this fascinating paper by Josh Karp from 2012 when he analyzed the same data set in how many? 4,000, 6,000 different ways, basically combinatorial exploration of this parameter space to see what difference does it make. And he did see that I think 90% of the cortical voxels were activated in at least one of those analyses. However, most of them did show roughly similar results. So we want to try to build tools that enable researchers to evaluate this effect on their data set without them having to manually click through SPM 6,000 times. And finally, we want to reach out to other communities. We acknowledge that there are a lot of smart people out there who do not have the expertise to deal with three-dimensional images or do not have the neuroscientific knowledge, but know quite a bit about data and signal processing. So all we need to really do is to extract meaningful features from the brain and provide this data and basically a set of dependent and independent variables to them in a form that they know and love, for example, a CSV file. So we will start doing that for OpenFMRI datasets and start setting up competitions for the machine learning community and see what the brightest minds out there can come up with. And finally, BITS is still evolving. We are working on a PET extension and MEG extension. We want to have a standard for describing models as well as derivatives, so outputs of an analysis. And I encourage all of you to get involved in that because the more voices we hear, the better the final product will be. So I want to thank my lab, Laura and John Arnold Foundation for paying me, and the INCF Data Sharing Task Force, and many people involved in all of these efforts. Without them, none of this would be possible. And on a final note, I want to introduce you to the OHBM Replication Award, which there's a submission deadline in, I believe, 21 days. The idea is that you can submit either yours or your paper with a justification and trying to promote replications in your imaging. So if you have a data set of your done a replication in the past, you can submit it for this award, and we acknowledge that replications are hard to publish, so those do not need to be peer-reviewed papers. All it takes is a preprint to be eligible for this award. So if you ever done a replication or you're sitting on some data that was acquired to replicate someone else's effect, you can write it up, submit it to a preprint, apply for the award, and bask in glory. Right. You can read more about this in those URLs. You can also Google OHBM Replication Award if you have any question. This hopefully will be an ongoing thing, so next year the same thing will happen. The only thing that's different this year is that you can submit papers or preprint that were published in the past, any time in the past. So if you published a replication or you published a paper which included the replication 10 years ago, you can submit right now, but next year we're going to limit that to a kind of a yearly basis. Okay, and finally, I'm going to leave this slide up. Those are different links and ways you can help and get involved. And I'm sorry I did overrun 5 minutes, but questions?